Okay, students. This one is mechanism of concentration of the filtrate. Okay, this is the diagram that shows the nephron and vasa recta nephron and the vasa recta, which shows the counter current mechanism. Okay, firstly, if you observe the loop of Henle, this is the descending limb, this is the ascending limb. Okay, urine, primary urine flows through the firstly loop of Henle. In loop of Henle, urine that flows through the descending limb from cortex region to the medullary part, and another one, the urine that flows through the ascending limb of the loop of Henle from the medullary part towards the cortex region. Okay, opposite manner in these two limbs. Okay, it is a downwards position, it is upwards position. That is called counter current. And if you observe the vasa recta, it is the, already you know about the vasa recta. Vasa recta means the efferent renal arteriole that forms the network of blood capillaries. These are called peritubular capillaries. Peritubular capillaries present at the region of the loop of Henle is called vasa recta. This is the vasa recta also has two limbs. This is locates just beside to the loop of Henle, also having two limbs, U-shaped structure like a loop of Henle. This is the descending limb, this is the ascending limb, but it flows the blood, not a urine. Vasa recta flows the blood, loop of Henle flows the urine. If you observe the descending limb, the blood flows from the cortex to the medullary part, and in this the blood flows from the medullary part to the cortex part of the kidney. Okay, this one is also the counter current. Opposite manner in two limbs, blood flows through the two limbs, that is also called counter current. Okay, the flowing of the urine and the blood through the loop of Henle and the vasa recta helps to maintains the increasing osmolarity towards the medullary interstitium. Okay, this is, for example, 300 milliosmolarity per liter concentration. 300 milliosmolarity per liter at the cortex part of the kidney. And if you observe the concentration, okay, this is in the medullary interstitium. This is equal to the 1200 milliosmolarity per liter. Okay, students, this is increases from the cortical part of the kidney to the medullary part of the kidney. Cortical interstitium increases from the cortical interstitium to the medullary interstitium, and the formation of the concentrated urine is also possible by the transport of the sodium chloride and the urea. Okay, firstly, what about the sodium chloride? sodium chloride that is present in the urine that flows through the ascending limb of the loop of Henle is entered into the medullary interstitium. From the medullary interstitium, this is absorbed into the blood of the vasa recta, through the descending limb of the vasa recta. Okay, NaCl present in this urine entered into medullary interstitium. From this enters into the blood of the vasa recta through the descending limb of the vasa recta. And finally, this is returned into the medullary interstitium by the ascending limb of the vasa recta. Okay, this one is about the transport of NaCl. Now, what about the urea? This is the urea. The urea present in the urine in the collecting duct entered into the medullary interstitium and absorbs into the urine through the ascending limb of the loop of Henle. From this entered into the medullary interstitium to maintain high osmolarity in the medullary interstitium that equal to 1200 milliosmolarity per liter. Okay, students, and this is the transport of the sodium chloride and the urea helps in much absorption of the water from the collecting duct to helps in maintenance of the formation of the high concentrated urine. Okay, finally concentrated urine is produces, this is entered into the ureter through the renal pelvis. Okay, students, now this is about the formation of the concentrated urine through the counter current mechanism. Okay, and micturition. This is the micturition means passing out of the urine to outside from the urinary bladder through urethra is called micturition. This is the micturition is by the involvement of the central nervous system. When the urinary bladder is fills with the urine, the stretch receptors, the walls of the urinary bladder having some amount of the stretch receptors, okay, goes to the stretch receptors present in the urinary bladder send signals to the central nervous system, and central nervous system send motor messages to the urinary bladder. Okay, now the urinary bladder, the muscles, smooth muscles present in the wall of the urinary bladder goes to the contraction and causes the simultaneous relaxation of the urethral sphincters. Now the urine is exit to the outside. The neural mechanism involved in the micturition that is called micturition reflex. Okay, students. Now this is about the micturition. Now what about the urine? Urine is light yellow in color. The pH of the urine is 6.0 and a normal healthy individual excretes 1 to 1.5 liter per day. Okay, And what are the contents present in the urine? It is 96% of the water and 2% urea and 2% dissolved substances. Okay, and this one is the urine helpful in examination of the metabolical disorders of the individuals. Okay, for example, it is helps to detect the disorder, that is diabetes mellitus by the presentation of the glucose in the urine, ketone bodies in the urine. Presence of the glucose in the urine called glycosuria. Glycosuria. Okay. Next, present of the ketone bodies. Present of the ketone bodies. That is called in the urine. That is called ketonuria. What about ketone bodies? Ketone bodies are formed by liver cells by the fatty acids. By the fatty acids. Okay. This is about the urine. Okay, students.